The fifth discovery on the list is that life is based on a digital information processing system. To explain this topic, I have to provide some basic background. So proteins are chains of amino acids that fold into three-dimensional structures. And there are 20 natural amino acids that make up proteins. The backbone chemistry is the same, but the side chains differ. Each amino acid can be represented by a letter, and so a chain of amino acids would look like this, a long chain of a combination of these 20 different letters. DNA is like a different language. It's a chain made up of a different kind of molecule called nucleotides. And there are 4 nucleotides that make up DNA. So it's a language of 4 letters. And so a chain of nucleotides, or a strand of DNA, would look like this. It's A's and T's and C's and G's in many different orders and combinations. So there are these 2 languages that are critical to all forms of life, and there's also a system for translating between these 2 languages. The genetic code maps each amino acid with a combination of 3 nucleotides. As shown on this chart, there are 64 triplet combinations of nucleotides that code for the 20 amino acids, or else for stop signals. The stop signals indicate the end of a protein in the translation process. This code is the essence of the system to translate between the 2 languages. But you might wonder, where does the code come from? And also, how can a code be written into molecules? We know the answer to the second question, so I'll cover that next. Above the chart are images of 2 complex molecules called tRNAs, or transfer RNAs. And if you look closely, you will see that each of these has 3 letters on one end. Those are 3 nucleotides, and an amino acid on the other end. tRNA molecules exist for every amino acid. So these tRNAs contain the code to translate between the 2 languages. But I'm sure you're wondering, where do they come from? But before I explain that, I want to give one last bit of background on the process of translation between these 2 languages. This slide shows the generic steps of transcription of RNA from DNA, and then translation of the RNA message into strands of amino acids that make functional proteins, such as enzymes. The translation step is performed by a very complex machine called the ribosome that will be discussed further in discovery number 6. That machine uses tRNAs in the process of translation to make a protein. So the tRNAs are critical. They're at the heart of the translation process. I'll explain briefly how tRNAs are made in 2 parts. This slide shows the first step for one particular organism. The main point is that tRNAs are made by cutting and splicing segments of RNA. tRNA splicing occurs in all 3 domains of life. In the 3 domains of life and in different organelles in eukaryotes, there are many different or variant tRNA structures and splicing processes. The clover leaf structure shown in this slide is the most common or generic structure. tRNA splicing means that RNA strands are first transcribed from DNA and then are cut and certain parts spliced together, all this done by enzymes, to construct the mature tRNA structure. This splicing process is similar to the topic discussed in discovery number 11 on the list, but the splicing of RNAs to make tRNAs involves an entirely different set of enzymes and processes. Critically important is that the loop at the bottom of the cloverleaf structure contains a triplet nucleotide sequence that will be paired with a specific amino acid to translate between the two languages. In the slide, those sequences are indicated as GCC, CCC, UCC, and so on. The information for those codons, the complementary sequences, were contained in the DNA strands that were transcribed. tRNA splicing is a very, very complex process. This slide provides references for review articles and a video on tRNA splicing processes. In the second part of the process of constructing tRNAs, the correct amino acid is fused to the end of each tRNA molecule. This is done by enzymes called tRNA synthetases. Generally speaking, there are 20 of these corresponding to the 20 amino acids. There are only 20 of these enzymes, despite the fact that there are 64 codons, because the code is degenerate. That means that more than one triplet codon in mRNA will code for the same amino acid. Triplet sequences that code for a specific amino acid vary in the third position. That's shown in the table given a few slides back. These critical enzymes implement the genetic code. They are universal, meaning that they're present in all forms of life. There are two distinct classes of these enzymes. This slide illustrates how these enzymes function. First, each enzyme will only bind one of the amino acids. So that means that it has a cavity with the correct physical and chemical structure to selectively bind only one of the 20 amino acids. The enzymes also contain binding pockets for ATP, a small high-energy molecule, which are structured in such a way that when ATP binds, a chemical reaction is catalyzed that activates the amino acid. Notice in the figure that each enzyme also has a cavity that selectively binds only tRNAs with the correct triplet codons that correspond to that amino acid according to the code. The enzyme is structured such that when the appropriate tRNA binds, a reaction is catalyzed that fuses or ligates the activated amino acid to the end of the tRNA molecule. So the code is written into this set of enzymes. But you might wonder, where do these enzymes come from, and how did it come about that they have the code written into them? The answer to that is that no one knows. Note that the enzymes are made by the same process of translation that they encode. So these enzymes themselves can't be made without the translation process, including the tRNAs that they construct. This is an example of an irreducibly complex process that requires many component parts to perform a critical function. If one of the critical parts is missing, the system doesn't function. So where does the irreducibly complex translation system come from? No one knows. Recall that discovery number 3 showed that the sequences of proteins that fold to make large domains characteristic of enzymes are extremely rare, like 1 in 10 to the 77th power. Now on top of that improbability, we have to include the improbability that a random sequence would fold to the correct shape to bind a specific amino acid and also the corresponding specific triplet codon sequence, and to catalyze the necessary reactions. Further, an enzyme of that type is needed for each of the amino acids present in the first life. I don't think any knowledgeable person would believe that happened by chance within our universe. And so most scientists would say that there must have been a simpler translation system at first that somehow evolved into the complex system that exists today. But the system of translation and the tRNA synthetases that exist today exist in all domains of life. There is no simpler version. This slide provides a couple links to some recent review articles on tRNA synthetases. A review article was written in 2009 with the title Origin and Evolution of the Genetic Code, the Universal Enigma. I really appreciate these authors because they state the issue clearly rather than sweeping it under the rug. That's actually rare in the literature on this topic, as I will discuss further below. I'd like to read a few paragraphs from this article. It says, at the heart of this problem is a dreary, vicious circle. What would be the selective force behind the evolution of the extremely complex translation system before there were functional proteins? And of course, there could be no proteins without a sufficiently effective translation system. He mentions a variety of hypotheses have been proposed to attempt to break the circle, but so far none of them seems to be sufficiently coherent or enjoys sufficient support to claim the status of a real theory. Near the end of the article, we read this: we're unaware of any experiments that would have the potential to actually reconstruct the origin of coding, not even at the stage of serious planning. Summarizing the state of the art in the study of the code evolution, we cannot escape considerable skepticism. It seems that the two-pronged fundamental question, why is the genetic code the way it is, and how did it come to be, that was asked over 50 years ago at the dawn of molecular biology, might remain pertinent even in another 50 years. Our consolation is that we cannot think of a more fundamental problem in biology. It's now well known and not controversial that life is based on a digital information processing system. Craig Venter said, life is a DNA software system containing digital information or digital code, and the cell is a biological machine full of protein robots. Richard Dawkins said the machine code of the genes is uncannily computer-like. Carl Sagan said the information content of a simple cell is around 10 to the 12th power bits, comparable to about 100 million pages of the Encyclopedia Britannica. And Francis Collins said that DNA is something like the hard drive on your computer containing programming. So to summarize, this discovery is that life is based on a digital information processing system. And I think it's fair to say that that was a huge surprise. 100 years ago, no one imagined that that would be the case. The code is built into a series of enzymes. No one knows where the code came from or how this information processing system could have come about. The system is irreducibly complex because many parts are essential for function. The digital information processing system at the core of life is a huge informational discontinuity. In discovery number 4, I mentioned this article by Eugene Koonin, who invoked the infinite universes explanation to account for the system, the digital information processing system at the core of life. I'll just read again one section of this that says, the origin of life is a chicken and egg problem. For biological evolution that is governed primarily by natural selection to take off, efficient systems for replication and translation are required, but even bare bones cores of these systems appear to be products of extensive selection, and he means by that they're very, very complex. He says eternal inflation offers a viable alternative that's untenable in a finite universe. That is, that a coupled system of translation and replication emerged by chance. That's a pretty extreme view, and I think few scientists would hold to that view or would admit it if they did. Most scientists, when speaking or publishing on this topic, would say that natural selection did this somehow. They attribute unlimited powers to natural selection without any evidential support. The review articles on tRNA synthetase enzymes mentioned earlier each have a section on the evolution of these enzymes. Taking a close look at those sections will provide a good example of how personal decisions are at the heart of this debate rather than scientific evidence. First, I want to point out that real progress in science is made not by imagining something, but by demonstrating something. There are an infinite number of scenarios that one can imagine, but only something that's true can be demonstrated. Imagining scenarios is often a helpful step in scientific discovery, but real progress in science is only made when something's demonstrated. The discussions on the evolution of tRNA synthetase enzymes in these and other review articles don't include any demonstrations of processes that could generate the information contained in those enzymes. Instead, it's simply assumed that natural selection did that. These sections contain phrases such as emergence, evolutionary history, traces of an evolutionary battle, evolutionary dynamic that shaped the code, and ancestral enzymes. Instead of a demonstration, the discussions involve imagining the order of steps in a hypothetical evolutionary process based on comparing the sequences of the enzymes for different organisms across the domains of life. The lack of any demonstration isn't surprising because natural selection requires a replicating system, and living organisms can't be used, since the simplest forms of life already have this information processing system. And if parts of the system are deleted, organisms aren't viable. Furthermore, there is as yet no such thing as a replicating, self-sustaining chemical system. So there seems to be no way to devise experiments to study whether it's possible for a chemical system to evolve a code in the tRNA synthetase enzymes. Koonin and his co-worker pointed that out in their review article. In principle, it's possible to design a computational system that could test such an evolutionary process, but as has been shown by William Dembski and his colleagues, evolutionary simulations to date smuggle in the information that's supposed to be generated by an unintelligent process. Please see the link on this slide for a discussion of that. To summarize, scientists who are committed materialists simply assume that natural selection can generate the enormous information content of the digital information processing system. They make no attempt to demonstrate that, but rather they just avoid or ignore the problem. Further, there seems to be no way to devise a system to experimentally test it. On the other hand, others recognize the huge informational discontinuities within the translation system that appear to be far, far beyond the capabilities of natural processes, including natural selection. It's like recognizing that a car on a beach couldn't have been produced by the repetitive action of the waves. It's obviously not possible. From this perspective, scenarios about the order of evolutionary events based on sequences ignore the elephant in the room, since they don't even attempt to address the informational discontinuity. If one does acknowledge that there is an enormous informational discontinuity, then the options are to believe in a designing mind, such as a computer programmer or a mind outside of the universe, or infinite universes and chance. A very basic question is this: can one ever question the limits of natural selection? Ever? Because if you do, that means that you must consider other causes. So experts in the field come to very different conclusions based on personal choices about what causes one is willing to consider, rather than the data of science itself. Finally, I'd like to ask you, which perspective do you think is more scientific? The one that recognizes the informational discontinuities, the limitations of natural selection, and the fact that minds can create information? Or the perspective that ignores the massive informational discontinuities, gives unlimited powers to natural selection while ignoring the known limitations of that mechanism? We will cover the known limitations of natural selection in Discovery number 7.